And so
Good. Hello, and welcome back from the break. We're going to go ahead and get started with plenary session number 4, clinical trial updates. I'll quickly introduce myself for those who I haven't met yet. My name's Kelly Brazo. I am the founder, along with my husband, of Cure LGMD 2I Foundation. Our daughter Samantha is 15 now, and when she was 2, she was diagnosed with LGMD 2I. Before it was known as also R9. And we were told at that time that, sorry, there's really nothing we can do for your daughter at this point. Keep her healthy, and if you want, you can try steroids. And my other option was to start a foundation because there wasn't enough funding for research at the time, and that was really the way to make a difference. So fast forward 13 years, here we are, and I know this is the moment that you've sort of all been waiting for, the clinical trial updates. I think it speaks for itself. This is really what everybody wants to hear today. So the landscape has changed dramatically from so many of the brilliant minds that are here in this room together, collaborating together, and we couldn't be more grateful. So we're all looking forward to hearing what you have to say today. So first, I'd like to introduce Doug Sproule from ML Biosolutions, and he's going to give us an update on BBP 418 clinical studies for LGMD 2I R9.
All right, so thank you for giving me the opportunity to speak at this conference today. It's a real privilege to be speaking on behalf of my colleagues at ML Biosolutions and our partners at Bridge Bio on an update of BBP 418, or clinical studies to treat LGMD 2I R9. Read this slide really quickly. No, BBP 418 has not been approved to treat any disease in any jurisdiction, and so keep all that in mind. Obviously, my disclosure, I'm the Chief Medical Officer at ML Biosolutions and carry that bias in everything that I do. ML Biosolutions was the product and emerged from the research efforts that were conducted at the McCall Lockwood Laboratory, and the discoveries that emerged from that lab actually have sponsored and led to the development of 2 companies. And this is a great example of how a philanthropy can turn a setback, tragedy in some people's minds, into something that can make a meaningful difference, not just in the lives of affected family members, but the broader community as well. And as a member of ML Bio, it's an immense privilege to have the opportunity to work in this space. We have rapidly progressed from an investigational new drug status in 2020 in just over 3 years, moving into phase 3, and it's an immense accomplishment of my colleagues, and it's a privilege to have the opportunity to represent them today. We will err, we will make mistakes, but at its core, Bridge Bio and ML Biosolutions exist to bring meaningful treatments to patients affected by rare genetic diseases, specific to ML Bio, limb-girdle muscular dystrophy type 2I. And we are passionate, and we have a commitment to fostering an open and transparent relationship with patients and their families. And we actively engage with the community in an attempt to help inform and optimize the design of our clinical trial protocols with the patient experience at the forefront. And it's been an immense privilege and an immense benefit, frankly, to us to have had the opportunity to have such a strong and collaborative relationship with a number of partners in the LGMD patient advocacy community and patients in general. I'm going to talk a little bit about LGMD2I, and it's for people who, for whom this disease has no relevance, and that's the majority of the people in this room. One of the awesome things that you're seeing in this session is the fact that we're moving from very little to multiple different companies having products in different diseases in the LGMD space, and this is an awesome thing. And so this is just a sign, hopefully, of things to come, and hopefully, we fast forward 5 years, there'll be dozens of companies, and you have to beat us all off with a stick trying to get on the stage. LGMD2I is caused by a mutation of a gene called FKRP, or fukutin-related protein. And this is a enzyme that is responsible for a single critical step in a process called glycosylation. And glycosylation is the attachment of this specific sugar chain onto the backbone of a different glycoprotein called alpha-dystroglycan. And so this glycosylation, this attachment of these sugar chains, are critical in allowing alpha-dystroglycan, or ADG, to function correctly. ADG is kind of thought you can think of it as a shock absorber for muscle cells during contraction. And if ADG doesn't work correctly, it's like the shocks on your car don't work correctly, chronic injury, chronic damage is going to occur, and that's what happens in LGMD2I R9. So one way to kind of think of it conceptually is to think of FKRP as kind of a water wheel in a mill. And so in the normal state, FKRP uses this substrate. In this analogy, it's the water flowing onto the water wheel. And in the normal, healthy, unaffected state, it's that water flowing onto the well is sufficient to drive forward and turn the wheel, and everything works well. We can think of it in the case of LGMD2I, where the FKRP enzyme doesn't work correctly. And so in this kind of analogy, the wheel is rusted. It can still sort of turn, but it doesn't really work right. And so that normal amount of substrate, that normal amount of water flowing over the wheel isn't enough to turn it. And so the mill wouldn't work correctly. BBP-418 is designed as a substrate supplementation. You can think of it as kind of flooding the zone. So we're flooding that well and putting a lot more water onto that wheel. And so even though that wheel doesn't turn correctly, if you give a lot of extra water, it'll turn it and make it work somewhat better. And that's the concept behind substrate supplementation. So BBP-418 is being developed as a substrate supplementation therapy. So it's designed to treat LGMD2I R9 at the source. So it's designed to increase the glycosylation of alpha-dystroglycan, increase in the amount of functional effective alpha-dystroglycan, which is the problem in LGMD2I. This drug is provided in the form of granules, so it's poured into liquid, it dissolves, and it's taken twice a day. So it's, drink it in the morning, drink it at night. In our clinical studies, I'll talk about this a little more, it's been well tolerated to date. There have been no dose-limiting toxicity, discontinuations due to drug, serious adverse events that have been observed related to BBP-418. And I'm going to talk a lot about the early clinical study results and some of the encouraging results we're seeing, particularly from our biomarker work and positive trends in functional outcomes. And I'm going to talk about that in the next couple slides. So my colleague yesterday, if you were attending the lunch and talked through our clinical program, but it's something that I think is a source of immense pride for me. We had a collaboration with our physician investigator colleagues in the GRASP consortium that enrolled approximately 100 patients in a natural history study through COVID. Some of you may have participated in that. It's an immense challenge to have done anything through that time, and that study has provided an immense wealth of additional data that I expect will be emerging in the scientific literature in the next coming years. We've performed 2 phase 1 studies that in healthy volunteers. This is to establish how we should think about the dosing, the pharmacokinetics, so how the drug is metabolized, how long it sits in our bodies for, and to look at some of the early safety and establish what is a safe dose in which to explore in patients affected by this disease. We have embarked upon a phase 2 study that's ongoing, and I'll talk more about that in a second. And we're really excited to announce our phase 3 study, which recently started earlier this summer in the United States, and we expect will expand overseas as well very soon, and I'll talk more at length about that in a few slides as well. Dr. Marks, in his remarks last night, spoke about the use of biomarkers, the use of secondary measures, of looking at biochemical tests as markers of whether or not a therapy, in the case of much of his conversation, gene therapy, is working to resolve and restore the underlying defect in a disease. And he talked about how this could be a possible premise that would drive forward an accelerated pathway to approving therapies in the rare diseases in general, and obviously very specifically the limb-girdle muscular dystrophies. And with that in mind, we've developed a bioassay that takes muscle tissue that's obtained by a needle core biopsy of the tibialis anterior muscle. So that's the muscle right in the front of the shin if you lift your foot up. And we use this to measure the levels of glycosylated alpha-dystroglycan, so the amount of functional alpha-dystroglycan in that muscle tissue. So what we see in patients who were in the natural history study, so these are patients who were not treated with any drug, is that the levels of glycosylated alpha-dystroglycan are markedly reduced when compared with a healthy control tissue that we use as a comparator. And you can see on this graph, the dashed line on the top is 100%. And on the left side are the patients who have the L276I homozygosity mutation. So these are patients who are the less severely affected population, although patients with that mutation I think would quibble with the description of their disease being mild. But those patients have a median of 90% reduction in glycosylated alpha-dystroglycan, so that's only 10% of what the healthy donor tissue would express. But that's still a lot more when compared with patients with other genotypes, which are typically much more severely affected. And for those patients, it's around 5% of the normal expression that we're seeing. And so this is kind of established that that this assay is able to reasonably be compared with what we know is the state of affairs and the clinical status for patients, and is a way that we could potentially track how these patients might respond to a therapy. We've utilized this in our phase 2 study. So this is a busy slide, but the simple way to kind of think about our phase 2 study is we had an initial 90-day dose escalation period. So there were 3 doses, different doses that patients received, and then all patients received 12 grams twice a day, which is our therapeutic dose that we're using in our phase 3 study. And we looked at a number of clinical endpoints, the North Star assessment for limb-girdle muscular dystrophies, ambulatory assessments, which is a 10-meter walk test, 100-meter time test, measures of breathing, measure of upper limb function, and measuring glycosylated alpha-dystroglycan levels in tissue, as well as serum creatine kinase, which, as everybody in this room hopefully is aware, is a marker of muscle breakdown that is obviously widely used in our field. This is a broad cohort. There were children, there were adults, there are patients who are homozygous for L276I mutation. There are patients with other genotypes. There are ambulatory patients and non-ambulatory patients. So small study, only 14 patients, but a pretty broad group of patients that we're seeing in this study. And the main goal of this study was to establish whether this is a safe drug to administer over relatively long periods of time in patients affected with this disease. And what we have seen is that across the range of the dose level study, we have not seen any serious adverse events that have occurred or considered related to BBP-418. And adverse events are undesirable or unexpected occurrences that affect the health of a clinical trial participant while that participant is taking an investigational drug. And so this is when, from a pharmaceutical development perspective, is obviously a critical feature when we're looking at whether a drug is safe, and then that allows us to look at whether a drug is effective. We did see 3 serious adverse events. They were determined to be unrelated to study drug. There have been no safety-related discontinuations. The adverse events that we have seen were relatively mild in nature, but that are deemed related to the drug include diarrhea, dehydration, nausea, vomiting, dyspepsia, gastroenteritis, and headaches. And they're generally short-lived and resolvable. We're really excited about the early results, particularly what we're seeing from the bioassay, from the tibialis anterior muscle biopsy samples, as well as the response we're seeing in creatine kinase, and that's depicted on the slide. So on the left side, you can see in the darker green are the patients who are homozygous for the L276I mutation, the less severely affected patients, and we're seeing a pretty robust increase in glycosylated alpha-dystroglycan levels from about 16% to about 40% of that normal tissue level following treatment with BBP-418, and that's sustained on several biopsies. Similarly, we're seeing a response in the patients with other genotypes. So those patients are coming at a much lower level, so around 5%, and we're seeing an increase to around 10% following treatment in those patients as well. And so we're also seeing on the right, you see change in creatine kinase, and we're seeing a large and marked drop in creatine kinase. CK is a marker of muscle breakdown, and we're seeing that this decrease sustained on repeated sampling. So we're looking at it on multiple times over an extended period of time. And so what this suggests to us is that there's evidence that the drug is doing something on the cellular level in the muscle cell, and that is translating to reduced breakdown of muscle. And we hope that will ultimately translate into clinical response, which we all really care about, right? It's great to have a lab test, but how does it translate to clinical? And that's going to be something that we're going to be looking at very, very closely, particularly as we move into the next phase of our clinical development. We are starting to see some early evidence suggestive that there is some clinical signal as well. On the red dashed lines are the patients in the phase 2 study. The blue are patients that were followed in the natural history study, so they're different populations of people. So that obviously creates a little bit of a confounder thing, so take this with a grain of salt. This was an open label study as well. But what we're seeing is that there's an improvement in the 10-meter walk test velocity, a reduction in the time to complete the 100-meter time test, which is in the middle, and an improvement in the North Star assessment for limb-girdle muscular dystrophies, which is shown on the right, when compared with the trajectory that we're seeing in untreated natural history populations. And so, all of this gives us enough kind of smoke, where there's smoke, there's fire, for us to push forward the program into a phase 3 study. And we recently initiated a phase 3 study. It's called Fortify, which will evaluate the safety and efficacy of long-term administration of BBP-418 in patients with LGMD2I/R9. And so this will enroll at least 81 patients aged 12 to 60 years of age. It's going to be a multi-center international study. It'll be a double-blind, randomized, placebo-controlled trial. I'll talk about that more on the next slide. And it's 36 months. It's a long study, which is the major burden that we are asking of the community. And we can talk about that. It's a function of the fact that stability is something that would be a major win. But to demonstrate that in using the existing outcome measures is quite a difficult thing. We are going to be opening what's called an open label extension study. And so all patients who participate and complete this study will have the opportunity, obviously it's always optional, to receive active drug through that study upon completion of this work. And we're going to be looking at a number of endpoints, including the NSAD, 100-meter time test, 10-meter walk test, pulmonary function, upper limb function. We're going to look at cardiac measures and other aspects, and we're obviously also going to be looking at changing glycosylated alpha-dystroglycan and CK as well. And certainly I encourage you to check out the clinicaltrials.gov, which has all the information that's publicly available for this trial and for every other trial as part of the regulations regarding pharmaceutical development. So to get into a little bit of detail, and still in a very superficial manner, there's a screening period during which a patient would meet with a trial investigator who would determine if a patient meets the requirements for the study. This would also be a time where the patient would be counseled and consented and informed of the benefits, the risks, the obligations of participating in a study, and would determine if he or she was interested in participating with the full information provided. Patients will then be randomly assigned to 2 groups. So 2 patients out of every 3 will receive the investigational drug, and 1 out of every 3 patients will receive placebo. It's a dummy, it will look and taste identical, and will be indistinguishable from BBP-418. And so this is double blind. That means that the study staff, the physicians, the patients, and us at ML Bio will be unaware of whether an individual patient is receiving the active drug or placebo, and this is to allow and remove the impact of bias and placebo and wishful thinking into how this product is assessed. Patients will be followed for 36 months with serial follow-up throughout, and that's the major obligation that this study unfortunately will have. And so we're really excited. This study is open already in the United States. There are a number of sites that will continue to open. We are hoping to be open in the UK and Europe before too long, and potentially also elsewhere in the world. And so it's obviously a very exciting time, and I'm looking forward to continuing to provide updates to the community. You know, we've accomplished a great deal for a tiny little company. We're excited to accomplish more as we move forward in partnership with the LGMD community. We've completed a natural history study in collaboration and in major effort of the GRASP consortium. We've completed 2 phase 1 trials. We have an ongoing phase 2 trial that I talked about. It's been presented at a number of conferences, and we'll continue to provide data as it emerges. But we've also been and will continue to be engaged with the community to get feedback on what's meaningful, what's important. We've conducted online surveys with individuals with this disease and their caregivers to collect feedback that helps us inform how we think about our phase 3 study. We've hosted focus groups. We continue to meet with patient advocacy leaders on a regular basis to get additional feedback and provide what information we're able to provide. And so we're very excited to see the next phase of things, and particularly to continue to engage with the community on how we can best partner and best move this program forward as fast and effectively as possible. And so I encourage you to visit our website for more information. Our email address is info@mlbiosolutions.com. By all means, lob us some questions and inquiries. We're happy to follow up. And thank you. I think the community has been an amazing partner for us. And we obviously can never fully acknowledge our appreciation of all the commitment and sacrifice that patients and their families have made, both as part of our clinical studies and the innumerable other ways that they are supporting the efforts to develop therapies for LGMD2I and for other limb-girdle muscular dystrophies. And we'd also like to particularly thank our advocacy partners for their continued collaboration and support. So with that, thank you, and I'll pass the baton back.
Thank you. And now I'd like to introduce to you Dr. Louise Rodino-Klapac from Sarepta Therapeutics, and she'll be providing you with updates on the LGMD program.
Hello, and thank you, everyone. I'm Louise Rodino-Klapac. I'm the Chief Scientific Officer and the Head of R&D at Sarepta Therapeutics. And I'd like to thank the organizers for giving the opportunity to talk about our pipeline, of which we have 6 LGMD subtypes in development. And I'd like to thank the organizers for organizing this meeting generally, because it's wonderful to see everyone here. I started working on LGMD research in 2005 at Nationwide Children's for 15 years, and then the last 5 years at Sarepta. And it's really wonderful to see a lot of friendly faces and new faces, and to see this community come together. So thank you for that. We also have forward-looking statements. You can find this on our website and read in more detail. We're a publicly traded company, and so I will be making statements that are looking into the future, and some of these comes with risks and uncertainties. So for the agenda for my presentation today, I'll first talk about why gene therapy for LGMDs, and all of the previous speakers really did a fantastic job setting this up. So I will talk about the relevant information, but now you are all little experts in gene therapy. Then I'll be talking about our SRP 9003 program, this an update on Study 101. This is for LGMD2E. But in general, throughout my presentation, what you'll see is that I'll be talking about specific programs, but we really have a platform approach. And so when I talk about data from particular trials, it really applies and is accelerating across our pipeline. I'll talk about our progress this year, and then looking forward into the years to come. And so why gene therapy? I think you've learned a lot about limb-girdle muscular dystrophy. Each is caused by an individual mutation in a different gene. All of them play some sort of role in muscle, whether it's at the sarcolemma or muscle membrane, which I'll talk more about, inside the cell in contraction or in muscle maintenance. And so improving this maintenance of the cell or the sarcolemma stability is important, and we'll get into details of that. But if you think about it like electrical wires, the cell membrane is protecting the muscle fibers and then the myofibrils, which are contained within each muscle fiber. And so you need that insulation to protect that muscle fiber, and without that, this can lead to limb-girdle muscular dystrophy. And so simply delivering a new copy of that gene is really treating the root cause of the disease. So I know this is a complicated diagram, but this just makes the point that there are many proteins present in muscle that are working together to stabilize muscle, to make it function properly. And when you have loss of one of these proteins within the cell, it can lead to a muscular dystrophy. So here I'm just going to give you one example of beta-sarcoglycan, which is the gene responsible for LGMD2E. And so when we're missing LGMD2E, this is an animation, you lose this entire protein complex. One of them is the sarcoglycan protein complex, and then that's part of a larger protein complex called the dystrophin-associated protein complex. And when that's missing at the cell membrane, what happens is that you get leakiness of the membrane. You get holes in the membrane, and over time, this leads to muscle cell loss. So the premise of gene therapy is delivering the gene that encodes for this protein to deliver it back so that you can restore this function, the protein complex, and the associated function. And that will be the theme you'll see throughout the talk. So let's start in an update of our ongoing study 101 for SRP-9003. And again, I'm going to talk to you about the mechanism of delivery of our gene therapy, and this really does apply across our platform. So SRP-9003 specifically aims to deliver the instructions for making beta-sarcoglycan in patients with LGMD2E or R4. This is still investigational, but we have trials ongoing which supports its development. So just looking at the construct itself, we use AAV RH74. So this is a version of AAV that is particularly good at going to muscle, and that includes skeletal muscle, cardiac muscle, as well as the diaphragm. And one of the first experiments I did back in 2005 was to look at RH74 versus other vectors, and this was selected. It was identified at Nationwide Children's, and we've been using it for quite some time now, and it's been very efficient at delivering to muscle. Next is the promoter. The promoter is like an on-off switch. So we deliver, I can show you here, we deliver through the vein, so just an intravenous injection. And so the virus, although it goes to muscle efficiently, it also goes other places like the liver. What the promoter does is allows it to just express and make that protein just in muscle, exactly where you want it. So even though it goes to other places, it's not making the protein there. And then the third component is the beta-sarcoglycan transgene, and then for our other programs, this would be switched out with the appropriate gene. So for example, alpha-sarcoglycan, gamma-sarcoglycan, et cetera. And so what we do, the day of gene therapy, it's a pretty anticlimactic day from what patients tell me, is that it's a simple IV, and it's delivered over the period of 1 to 2 hours. And what it does is goes to the bloodstream, hones to muscle, and then is delivered to the nucleus. And as you heard earlier, it doesn't affect the chromosomes within the nucleus of your cells. It just stays there as an episome and continues to make protein over time. So our first study, our 101 study, is a fully enrolled study. We enrolled 2 cohorts of LGMD2E patients, a low dose and a high dose. But all patients had to have confirmed mutations in LGMD2E, be negative for antibodies to AAV RH74, and be greater than 40% of normal on their time test. We had a wide age range of 4 to 13 years, which we've since expanded, and I'll talk more about. So the study was primarily focused to look at safety, but then also expression of the beta-sarcoglycan protein at both day 60 and 2 years. And patients are followed for up to 5 years in this study. So these are representative examples from both the low dose and the high dose cohorts of the biopsies. So these are looking at staining of beta-sarcoglycan. It's a cross-section of muscle. And so what you're seeing on the far left is a control. This is an individual without LGMD2E, and so you see beta-sarcoglycan surrounding each muscle fiber. And then if we look at the two cohorts on the right, so each patient has a baseline biopsy where you can see the absence of that protein, which is then restored at day 60 in both dose cohorts. And moving forward for trials, the high dose was selected to move forward for the ongoing studies. So why is it so important to look at expression? And Dr. Mark's talk yesterday really set the stage for this. We are proposing that beta-sarcoglycan is used as a surrogate endpoint. It's the sole cause of the disease, and so restoring expression of this gene and corresponding protein is really instrumental in a therapy. So as I mentioned, as we restore the protein, we're restoring the protein complex that's associated with it. And we can measure this, and we can measure the components of the complex very accurately. And we've shown in animal models, as well as clinical studies, that restoration of this complex is associated with corresponding function and a therapeutic effect. And so surrogate endpoints have been approved by FDA for use for the accelerated approval pathway, and this is the pathway that we're using for these LGMD programs, and I'll talk in more detail about that. And so we've looked at restoration of the sarcoglycan complex in great detail in this study, and so what we're looking at is the two different cohorts, and I'll walk you through the diagram because I know that they can be complicated. So if we just look at the diagram on the left, what we're showing is baseline expression and then day 60 and year 2 expression of the sarcoglycan complex proteins. And so we're looking at beta-sarcoglycan, delta, alpha, and gamma. And what you can see is that you see an increase in the amount of protein for each of these complex proteins that's not present at baseline. If you look on the right, this is just the cohort 2 where you see the same phenomenon, but just an increase in the amount of expression of that protein. And this is looking at positive fibers. So not only are we delivering the gene that is not functioning, but we're improving the amount of protein at the complex. And so this really validates beta-sarcoglycan as a surrogate endpoint for approval, for potential approval moving forward. We're also looking at a variety of measures of physical function. So the primary in this study is the NSAD scale. The NSAD, as you've heard, is a scale, modification of the NSAA scale that was developed to look at LGMD, particularly in both ambulant and non-ambulant individuals. It includes a scale of 29 different items for a total possible score of 54 points. And essentially, if you have a 0 on the scale for a particular activity, you're unable to do it. If you have a score of 1, you can do it with some modification, and 2 means you can do it with no modifications as normal. And so adding up this scale correlates to activities of daily living. We also looked at a number of time tests as well and monitoring pulmonary function. So this is the functional data for the NSAD scale. We've now looked for up to 3 years in cohort 1 of the study, which is the low-dose cohort, as well as the high-dose cohort. And what we found is an improvement in the NSAD score, which is then sustained over time. And so we'll continue to follow patients, but we're seeing nice improvement in stability of function over this time period. In terms of safety, in this trial and most gene therapy trials, the serious adverse events occur within the first 90 days of therapy. We've seen similar results here. We've seen side effects of nausea and vomiting being the most common, as well as liver enzyme elevation. We've had no participants drop out of the study because of side effects. So I'm going to go sort of one by one. We have, as I mentioned, 6 programs in LGMD, and I know that you're eager to hear where we are with each one of them, so we'll go through them in some detail. So in 2023, we also started a second study in LGMD2E called Voyaging, and this is a study to look at an expanded population of patients, both ambulant and non-ambulant, and I'll talk more about that. The second study is a study called Navigene, and this is in dysferlinopathy, or LGMD2B. This is a small proof of concept study to look at safety and expression of a dual vector construct for dysferlin. And we're excited to say the study's enrolled, and we're awaiting the outcome of that study. This third bullet cannot be understated, and I know that it's a bullet on a slide, but it's really a big part of this, and this is process in our manufacturing for all of our LGMD candidates. And so over the last several years, we've been developing a new process for manufacturing for all of our LGMD candidates, which is scalable and is our intended commercial process. And so what that means is when we go into the next limb-girdle trials, we'll be using the intended commercial process. We won't have to repeat studies by using bridging from one process to the other. So all of this work was really critical in getting to this point. Next, for LGMD2C or R5, which is gamma-sarcoglycan, we've identified some potential issues in the highest doses in our preclinical studies, and so now we're doing some additional non-clinical studies to be able to make sure that we have the right dose prior to initiating studies in humans, and I'll talk more about the timelines at the end. And then Journey is our natural history study in the sarcoglycanopathies. We've enrolled 137 patients, and that is a 3-year study, and I have a slide to go in a little bit more detail. So first, just looking at Voyaging, again, this is a second phase 1 study in LGMD2E, looking at ambulatory and non-ambulatory patients. It'll enroll 6 patients from a wide age range, from 4 to 50 years old. The study has been fully enrolled and look at biopsies at day 60, and then look at safety and functional measures over time. So all of these studies will be important for delivery of a package to FDA moving forward. So next is Navigene, and this is SRP 6004. Sorry for all of the acronyms, I know there is a lot of them, for dysferlinopathy or LGMD2B. As was mentioned earlier, dysferlin is a large gene. It's too large to fit into AAV. And so I developed a solution some years ago. We're delivering it in 2 different pieces, as Dr. Wicklund mentioned earlier. So the study is, again, looking at safety for up to 5 years, and then expression at baseline, day 90, and 2 years. And so just to show you a diagram of this, we actually deliver 2 different vectors at the same time. Through the circulation. We need 1 copy of both of them to get into the nucleus. And what we do is once they're in the nucleus, we're harnessing the power of the cell to use its own machinery to repair the gene and create a full-length transgene. So we have the MHCK7 promoter, that's that on-off switch in muscle, that's then expressing the full-length transgene. We've shown this repeatedly in animal models and early clinical work that we can restore full-length dysferlin, and so next steps we'll be looking at the biopsies in these patients and then setting up the next trial based on that data. As I mentioned, we have a Journey natural history study that many of you in this room have participated in. This is for all of the sarcoglycans. And we're following patients for over 3 years, and this is to look at various measures of physical function, pulmonary function test, and then exploratory measures involving cardiac and patient-reported outcomes. These natural history studies are really critical in informing our design of our clinical trials, and then also to show efficacy. And these are important, as Dr. Marks mentioned, to the FDA as well. So in terms of timelines, I'm going to go through each of the programs again just to touch on where we are with each program. So for LGMD2E, as I mentioned, we've initiated 2 phase 1 studies. We're on track to initiate a phase 3 study in late this year, and that study will be used using the intended commercial process and used for registration, or that will be submitted, the study that's submitted for approval. We are working on improving our manufacturing yields. We've developed a new process, and this allows for scalability so we can make more of these vectors at once. We can make them more efficiently, and then use just one process moving forward in single trials to help accelerate the development path. For LGMD2D, R3, we've been working on the manufacturing and then additional preclinical studies, and we're set to initiate a phase 1 trial for LGMD2D next year in 2024. For LGMD2C, as I mentioned, this is our 9005 program. We're doing some additional non-clinical studies to make sure we understand the safety profile before entering the clinic, but we're expected to start our phase 1 study in early 2025 for LGMD2C. We have 2 additional programs, which is LGMD2A and LGMD2L. These are marked in the preclinical stage, and this is where we're doing the manufacturing process as well as additional non-clinical studies. I heard you yesterday that the preclinical time sometimes seems like a very long time, and what I can say is there's a lot of work going on behind the scenes during this time to make sure that we have the process right, make sure that we have the safety right. But once we exit this process and go into the clinic, things start to accelerate, and all the work that we're doing across this platform is really helping to accelerate the next programs. So just to put the LGMD2E pipeline in a little bit more detail here, we've now enrolled these 2 studies, 101 and 102. And then the third study, 301, is our phase 3 study, which will begin at the end of this year. That is a global study in both US and Europe in both ambulatory and non-ambulatory patients. And this study will be used as a registrational study. So I think it's important to highlight our regulatory approach. I mentioned this, and Dr. Marks talked about it in detail. Really, the limb-girdle muscular dystrophies are set up well for the accelerated approval pathway. We know the gene that's missing. We know that we can deliver that protein. We know in animal models and clinical data that restoring that gene or protein can lead to functional benefit. And so using this accelerated pathway, making sure that we have safe therapies that express the protein are important. But doing long traditional functional studies is very difficult in slowly progressing diseases. So we are choosing the accelerated approval pathway to move forward, and that will be for all of our limb-girdle programs. To show that the protein is reasonably likely to predict clinical benefit, and then we will be following for functional measures over time to convert to traditional approval. So this is our pathway moving forward for across our pipeline. So as a community, we're here and doing this for you, but we couldn't do it without you. Everything that you do as a community, don't underestimate how powerful your voice is and your contribution to studies. So there are many ways that you can get involved and engaged in addition to what you're doing now. So you can advocate on issues. You can participate in surveys. Surveys are really important. Dr. Marks talked about this yesterday. Understanding what's important to the patient community in terms of potential outcomes, what you see as a success in treatment is really critical for us to know, for the regulators to know. Participating in natural history studies is critical as well for potential contributions to future gene therapy studies, as well as just understanding the natural history studies and the outcomes. If you haven't, get genetically tested. For gene therapies, it's absolutely critical to know what your particular subtype is, because there are corresponding gene therapies in development for those. Advocate for your ICD-10 code. This was mentioned yesterday. This is also critical for giving each subtype the acknowledgement that it needs moving forward and to stay connected with therapies. We have many ways for you to, I think you've all probably seen the Sarepta team, to reach us. Find us advocacy.sarepta, limgirdle.com. There are many ways to reach out to us. So just like to stop by thanking again for the Speak Foundation for giving us the opportunity to speak today. I'd like to thank patients, the caregivers, clinical trial sites, healthcare providers. It's all critical on our journey to develop treatments for limb-girdle. And I know I had to go through a lot of that fast, but we will be having lunch after this, and you are welcome to come up and ask any additional questions. But thank you.
Okay, so we have one more talk before lunch, and I'd like to invite Dr. John Vissing to the stage from the University of Copenhagen, and he's going to be speaking about the preliminary experiences with Atamyo's FKRP gene therapy.
Thank you, Kelly. Thank you for having me. I'm really happy to be here. It's a really nice meeting. So I will be talking about the very early stages of a gene therapy program that we are currently doing in limb-girdle muscular dystrophy type 2I or R9 fructin related, FKRP related. So these are my disclosures. They're a little bit more easy to read than the forward statements, but still very boring, so we are gonna go on and jump right to it. So as Dr. Sproule already alluded to, FKRP is important in putting sugars on the self-membrane of the muscle cell, and this may confer some mechanical stability, as we have heard, but it may also be important for cell signaling, so that outside cells can recognize the cell. So this is very important. So to try to make a gene therapy for this condition, Isabelle Richard at Genathon in just outside Paris in France tried to do this starting some 7 years ago. And she constructed this, the gene. It was a complete human FKRP gene, which was optimized so that it actually expresses more FKRP than the one healthy people carry. And this was put into an AAV9 virus. The reason for choosing this virus is that this virus is known to penetrate muscles very well. And in the first go around, a lot of things went right, but the mice that they were treating had cardiac toxicity. So too much FKRP. We have FKRP in the heart, but too much of it confers cardiac toxicity, so they had to go back again in the lab and again modify the gene so that it would not express in the heart. And the next slide really, and I'm sorry this is really busy, I promise you this is the most complicated slide I'm going to show you, but I think it's important to show, all the preclinical work, and this really recapitulates work, 4 or 5 years of work. So to the left, you see the histology slides from the mice. The top one there is untreated, and you see some of the weak staining in some of the muscles there is because the muscle cells are dying. And at the very bottom, you see a healthy mouse. And then in between, you see 3 other slides. And these are mice that have received the gene therapy. And you can clearly see that even at the low dose, there's a nice restoration of the histology. And the next, the graphs here show you the 2 gene constructs. The first one in blue, the one that is expressed in heart, and the red one, which is the new gene construct, which is now used in the gene therapy trial in humans. And this shows you that the gene is expressed in muscle. And highly so in the heart in the first gene construct, but the second gene construct you can see in the heart there's no expression at all. And then it also helped the mice in their histology. You can see that in the middle graph there's a high level of centronucleation. And what does that mean? That means that the muscle is damaged and it's trying to repair itself. So it's a sign of muscle damage, and when you then treat the mice there in blue and red, you see that essentially they are almost as the healthy ones, which is the green right to the left. And then the muscle strength, which is the last graph here on the right side, you see that in the black one there's a dip in the force. These are untreated mice. So they have decreased muscle force like we have in humans. And with the treatment, which is the 2 blue and red here on the right, you essentially restore muscle strength in the mice. So this was quite promising, and therefore this moved on after some 5 years or so work in the lab to the clinic, and now we have then started this gene therapy trial in humans. And it comes in 2 stages. So the first stage is a phase 1 study, which is now ongoing. That's the top of the graph. You can see there that it's a dose escalation study. So there are 2 doses, and we have completed the first 3 patients at the low dose. They have been dosed, and we are now starting the higher dose. The patient was screened on Wednesday and will probably be dosed the week after next at the high dose. And this study, this phase 1 study, is only carried out at 3 sites. Professor Volker Straub, who you heard talk yesterday, has a site in Newcastle upon Tyne in the UK. And then there's Dr. Tanja Stojkovic in Paris, and then our site in Copenhagen. And then the second part, if everything goes right, and it looks as if things are on track right now, we will then move on to the phase 2, which is the bottom of the graphics here. We will include many more patients, 33 in total. A third of the patients will receive placebo to begin with. They will be followed for 1 year, and then the placebo patients will then receive gene therapy. So eventually everybody will receive gene therapy in this program. And in the phase 2, there will be more sites, and I know there will also be some American sites. They have not been picked yet, but will be picked by the sponsor, Atamyo, soon. So the inclusion criteria for the trial is that you have to have a lung function which is not too bad, but it should be affected. So a functional vital capacity below 80, but more than 40%. And you should be able to walk 10 meters in less than 30 seconds and be able to rise from the chair. For the phase 2, this will very likely change. So you have the same lung criteria, but you also need to have a North Star below 40. And the reason really for that is based on findings in natural history studies. So this is not the gene therapy study. This is now a natural history study of 52 patients that was conducted as sort of a run-in for the gene therapy trial. And here, when we divide the cohort in the total cohort, patients that just have a lung function below 80% of normal, and patients that besides this lung function also have a North Star below 40, you can see when we look in a 1 year time that the lung function drops more in the more severe group. Out to the right, the 10-meter walking velocity drops more as well. The time up and go also is more affected. It should be a minus there with in front of the 3.7. And the same with the North Star assessment. So it looks as if a slightly more severe phenotype progresses more, and it's easier to show effect in such a group. So it will be the main target for the phase 2 trial. The primary endpoint right now is functional vital capacity, because we've shown that this changes quite a bit in natural history studies, but probably it will be a combined FVC and North Star assessment, which will be the primary endpoint in the trial, and FDA has requested this. But there are other important endpoints. Patients undergo muscle MRI, and a number of functional test biomarkers will be FKRP expression, also muscle glycosylation, of course, and CK. So What is the safety? We've dosed 3 patients, 2 Danish patients and 1 French patient, and the 2 Danish patients have, one has had the treatment 1 year ago, and the other patient half a year ago. So there were no serious adverse events. There were some adverse events related to the corticosteroid treatment with nausea, vomiting, and so on. No cardiac affection. Actually, one of the patients had a cardiomyopathy and had even a drop in their pro-PNP, which is an indicator of affected heart function. So that was quite positive. There were some elevated liver enzymes, and these were from the liver and not from the muscle, which were probably related to the viral load, which builds up. For many gene therapies, you can avoid getting a lot of virus in the liver. So in the beginning, this is why we give immune suppression to the patients in the beginning of the treatment, and they have resolved all these problems. And in the 3rd patient that was dosed in August, there were really no adverse events at all. So this is the creatine kinase or CK levels in the 3 patients that we have dosed so far. We see that there is quite a marked drop in the CK levels. To begin with, these patients are on corticosteroid treatment, which in itself could decrease the levels. But what I think is remarkable is the 2 patients, the blue and the red, as we come out to, after 3 months, these patients are then not on immunosuppressants and the CK levels stay down. CK is a terrible biomarker for most studies anyway, but I think it is remarkable when it almost becomes normal. And that is the case in these patients. So very encouraging. Also, there were some improvements in the walking velocity, mostly in the 2nd patient we dosed. Mind you, these results are open label, and there can be some placebo effect, obviously, in these patients. They know they're getting the gene therapy. Also, some improvement in the quality of life, mostly in one of the patients, and stabilization in the others. And they report disappearance of myalgia and cramps, which is positive. And if we just take a quick look at the 2 patients that had the longest duration of treatment, so the table on the left there is the patient that had a treatment for 1 year. We can see overall there is an improvement in everything. The column to the right shows the natural history data, what we should expect. And basically everything moves in the opposite direction in this patient. The same is true for the patient who we followed only for 6 months. We don't have all the assessments yet, but it also looks quite promising. And then what about biomarkers? So one thing is, as I showed you in the mice, central nucleation in the muscle is a sign of there is a regeneration going on, and of course that's good, but it's also a sign that there was damage. So if you have less central nucleated fibers, that's a good sign, and as you can see in the 2 patients that had a biopsy at 3 months, the number of centronucleated fibers, as we saw in the mice, drops. And interestingly, when we look at the FKRP expression in the muscle, and if you can, on the picture on the left, you can see these red, teeny tiny small dots. That is the FKRP expression, and to make sure that this is really a massive expression, we only count the fibers that have at least 29 of these spots, and those cells are then on the right side depicted here in red. And this is quite comparable to what we see in in the mice studies. So quite reassuring. And obviously we will also be looking at glycosylation in these patients, but we don't have these results yet. But this is encouraging. So to wrap up, so the safety profile looks quite good so far. They have had some adverse events, some affection of the liver, not that they had any symptoms, the patients, and it resolved, so nothing serious there. Also the efficacy data that I showed you, but be mindful of this is open label, still early days, difficult to interpret, but it does look good and encouraging. And it also looks on the biomarker that we are actually getting the gene to work in these patients. And as I already said, you know, the data safety monitoring committee approved moving on to the higher dose just earlier this month, and we will be dosing the patient at the higher dose, which is a 3 times higher dose than the lower dose, in just a week or so. Is this a cure? It's not a cure, and I heard you know, a lot of people talking about a cure, and I think you can potentially cure someone if they're pre-symptomatic and you can transfect all cells in the body, all muscle cells. Then I think you can talk about a cure. But obviously, people who are treated here have had the disease for quite a while, and gene therapy doesn't, you know, make new muscle cells. It treats the cells that are there. So just to be mindful of, you know, manage the expectations here. I'm not saying that it's not really important, because it could be a really important treatment, but it is not a real cure in that sense. And obviously, there can be muscle cells that are not transfected, who will not get the gene therapy, and they will still be ill. So, but it looks as if it could be a great step forward. And with that, I think I will finish.